Hi, so for our senior project, we worked on developing a nanoladder scaffold for neuronal outgrowth and regeneration. So the big issue that we're tackling are spinal cord injuries, which affect roughly 253,000 people in the US alone, as well as an additional 11,000 each year. Unfortunately for those suffering from spinal cord injuries, there are only palliative treatments. Some of the main causes of spinal cord injuries are violence and bullet wounds, vehicular accidents, sports accidents, and falls. So the big issue that we're tackling are addressing the barriers that spinal cord injury healing. So the first big barrier is called the secondary injury, which is a cascade of biological events that occur at the site of injury after the primary injury that include increased cytotoxicity, tissue loss, and, scarring, as well as inflammation, which make it really difficult for tissues to heal and regenerate. And the other barrier that we're addressing is that there's no inherent directional guidance for the neurons to grow and reform their functional connections in the body after a spinal cord injury occurs. So there's been some previous work done to study how we could regrow neurons and reform functional connections, and a lot of them have to do with developing scaffolds that look at both topographical and chemical features. Topographical features generally span on the micro and nano scale, and chemical features are generally looking at proteins and things like that that stimulate growth. So that brings us to our goal for the project. So our goal is to develop a nanoladder scaffold that would both regenerate neurites and elongate them, but also give them directional growth guidance so that they could grow in the proper direction when implanted for reestablishing their functional connections and establishing function to the body. So there are three central aims for our project. The first was to study different extracellular matrix proteins, and we compared neurite elongation on those different proteins. So we basically wanted to see which chemicals and which proteins would elongate neurites the best for our final project, and so we would know which ones to incorporate. So the second central aim was to actually develop the scaffold. So previously in our lab, they didn't use silk as a substrate, but we decided during the middle, roughly in the middle of our project, that we were actually going to move to silk as the biomaterial that we use for the scaffold because of its biocompatible nature. But that meant that we had to establish a protocol for actually fabricating the nanoladder and the micro scale and nanoscale features on the silk substrate. And I'll talk a little bit about the fabrication process in a minute. So the third critical aim for our project was to evaluate the neurite elongation on the silk nanoladder, because we wanted to make sure that the cells would be able to grow and adhere to the silk nanoladder, and we wanted to make sure that the silk didn't inhibit growth for any reason. And we wanted to just basically compare that to our previous results. So for our approach for our one of our central aims was the actual fabrication of the nanoladder. So that started with a cover glass slide, which we added a layer of photoresist to. And then we used photolithography to strip away micron-wide strips. And then we adhered polystyrene beads to the nanoladder structure. And then we coated the cover glass slide with a silk solution, and then we let that sit overnight. And then we peeled off the silk film, and it had imprinted on it the micron-wide stripes, as well as the grooves that we got from the nanoparticles. For our approach for the ECM protein coatings, we started off with sterilizing our silk scaffolds, and then we prepared ECM protein solutions. So we looked at laminin, poly-D-lysine, and fibronectin. So we coated the scaffold in ECM proteins, then we cultured the cells on them and incubated them overnight. For the third central aim, which was the actual cell culture and imaging, so like I mentioned before, we used embryonic spinal cord cells, seeded them on the scaffolds, and incubated them for 7 to 10 days, and then we used anti-tau staining and imaged them with confocal microscopy, and we used an ImageJ plugin developed by our lab to study both directional alignment and also length. So data for the protein coating showed that laminin was significantly better at enhancing the neurite elongation than both poly-D-lysine and fibronectin. and a t-test found significant differences between all groups.